Studies on crude glycerol degradation and hydrogen production by photofermentation. Abstract hydrogen is considered as an efficient green energy carrier that can be produced in and by biological methods. Photofermentation is one of the biological methods that is said to be effective for biohydrogen production. The bacteria used in the process are facultative anaerobes that are also capable of utilizing waste organic effluents from various process industries as carbon substrates. The current study was based on use of crude glycerol, obtained as a byproduct from biodiesel plant, as carbon substrate for the photofermentative bacteria, Rhodobacter meghalophilus, for hydrogen production. The inoculum size and the light intensity was varied to study their impact on the bacterial growth and hydrogen production process. The inoculum size was varied at 1 ml/L, 2 ml/L and 4 ml/L of the fermentation medium, and lead lights of 5W, 10W, and 15W was used to supply light source needed for photofermentation. Increasing the inoculum size was found to aid in better degradation of crude glycerol, but 2 ml/L of inoculum size was found to be effective for hydrogen production. Use of lead light source showed that 10W was sufficient for photofermentation, as light intensity at 15W was found to inhibit the bacterial growth and did not help in to increase hydrogen production. Thus, highest yield of hydrogen gas was obtained with 2 ml/L of inoculum size and light capacity of 10W. These results showed that inoculum size and light source are among the crucial parameters governing hydrogen production by photofermentation. Keywords: alternate fuel source, biohydrogen, photofermentation, crude glycerol. Introduction. Man's strive for modernization and development is leading to rapid drying up of the oil wells, which is being a major source of energy boost for all technical advancements happening around us. To meet our energy demands and to sustain the development, the urge to provide an alternative solution has become an utmost necessity. Of the many alternative sources being explored for energy, hydrogen is considered as a versatile replacement for energy. The calorific content of hydrogen is considered to be higher, which makes it a promising substitute and a highly reliable energy carrier to meet most of our energy needs. Hydrogen gas has many industrial applications in production of ammonia, petrochemicals, and is also extensively required in refineries. 1. Steam methane reforming, partial oxidation of hydrocarbons, 2. Coal gasification, 3, etc., are the major producers under the chemical method. 2. Biological method could be more advantageous for hydrogen given its low production cost, versatility to use a variety of feedstocks, and eco-friendly. Biological methods such as photofermentation, dark fermentation, anaerobic fermentation, 2, is studied extensively to optimize the production process. The bacteria employed in these processes can derive energy from many hydrocarbons such as glucose, sucrose, maltose, 4, etc. However, these bacterial sources can also feed upon many waste organic materials, rich in hydrocarbons such as agricultural residues, 5, food wastes, 6, industrial organic effluents, 7, etc., and thus help to make the process economical and minimize the potential hazards that arise due to disposal of these organic materials. Biological processes require mild to moderate process conditions such as pressure and temperature, making it economically stable. Unlike chemical processes, biological process produce only water as its byproduct and thus, is more eco-friendly. These qualities of a biological process are advantageous over chemical route, and therefore, hydrogen by biological processes is being explored as part of the green energy technology. 2. Literature survey. 1. Photofermentation. Biological routes mainly consist of direct and indirect photolysis, photofermentation, anaerobic fermentation and dark fermentation methods. Among these, dark fermentation and photofermentation are the most preferred process, and are often carried out as a combined process given their increased efficiency and does not involve any complicated system design. 8, 9. Photofermentation involves anaerobic process carried out in the light by purple non-sulfur bacteria. Dark fermentation is a simple, anaerobic process that decomposes the organic matter in the absence of light. The process can be carried out at room temperature and pressure. The potential of both the processes to incorporate a wide variety of organic compounds as its substrate has drawn most researchers to explore the use of diverse industrial organic effluents for biohydrogen production. 10, 11. The key aspects of fermentation for hydrogen production are: it is inexpensive, has lesser maintenance, simple bioreactor designs, and capability to incorporate a wide range of waste organic materials as substrates. These factors are advantageous compared to chemical methods such as steam methane reforming or autothermal reforming that are energy-dependent processes and also work on the fossil fuel feedstocks. On the other hand, Some bottlenecks that still impair the scale-up process of biological processes are its low stability, separation of hydrogen from biogas, formation of byproducts such as organic acids and alcohol, low energy efficiency of the process, etc. 12. 2 microorganisms for photofermentation and dark fermentation. Hydrogen-producing microorganisms are said to be either facultative or strict anaerobes. Microorganisms employed for photofermentation are facultative anaerobic bacteria such as Rhodobacter sphaeroides, Rhodospirillum rubrum, Rhodobacter capsulatus, 13, 14, etc. while bacteria employed in dark fermentation are anaerobic, fermentative bacteria belonging to the species Clostridium butyricum, Enterobacter aerogenes, 10. These bacteria are a few among the potential microorganisms that are suitable for hydrogen production. While these organisms can feed on a range of monosaccharides such as glucose, sucrose, maltose, fructose, etc., as their carbon substrate, it is crucial to select the bacterial species that are capable to acclimatize to the type of waste organic material, used as carbon substrate for hydrogen production. Waste effluents released from many process industries are rich in carbon matter. Agricultural residues are also rich source of carbohydrates and thus, these waste sources can be used as potential feedstocks in biological hydrogen production processes. Effluents from breweries, 15, beverage industries and food processing plants, 16, 17, 18, dairy units, 19, etc. and agricultural crop residues such as wheat, 20, 21, 22, potato, 23, 24, molasses, 25, etc. are being considered as feedstocks. Biodiesel, one of the alternative fuels, finds extensive use in transportation sector, as a blend with conventional diesel. Biodiesel production plants are being commercialized for large-scale production. Biodiesel is produced by trans-esterification of vegetable oils and fats using solvents and catalyst, where glycerol is removed in the process. This is collected as byproduct that accounts for 10% of biodiesel production. Solvents such as methanol and sodium salts, used as catalyst are also obtained along with glycerol in this byproduct. Glycerol can be converted to many value-added products such as 1,3-propane-diol, 26, propanoic acid, and butanol, citric acid, ethanol, 27, etc. As glycerol being a high-carbon source, it can also be used as feedstock for hydrogen production through biological processes, 28. 3 materials. Crude glycerol used as substrate in the current study was procured from the Biofuel Park, Gandhi Krishi Vignana Kendra, University of Agricultural Sciences, Bangalore. The chemicals used for the medium preparation was procured from SRL Chemicals of AR grade. The culture of meghalophilus was obtained from Jawaharlal Nehru Technological University, Hyderabad. The bacterium was first subcultured in the growth medium, as per the composition defined in Rhodospirillum medium, modified, DSMZ medium 27, with 0.3% sodium pyruvate. 4 methods. The free glycerol in the crude mixture was determined by a reaction of crude glycerol with sodium periodate that forms formic acid. This is then reacted with acetyl-acetone that forms 3-diacetyl 1,4-dihydrolotidine, which is a yellow-colored complex. The intensity of this yellow color is directly proportional to the glycerol content. The absorbance of the colored sample was measured at a wavelength of 440 nm and the concentration of glycerol was determined from the calibration chart for glycerol. Glass reagent bottles of 1000 ml capacity fitted with stopper, was used as reactor. The stopper was fitted with nitrogen flushing tube and gas collection tube. The outlet of the gas collection tube was inserted into an inverted cylinder, placed in a water-containing trough. The gas produced during the fermentation process was collected by volume displacement method. The reagent bottle was placed on a magnetic hot plate and photofermentation experiments were carried by supplying light energy. As photofermentation occurs in the presence of visible light, LED bulbs were used to supply the required light energy. Meghalophilus is a mesophilic bacterium and the process temperature was maintained at 30 degrees Celsius. Crude glycerol is viscous in nature and the presence of fatty acids or the esters make it immiscible with the medium components. To ensure uniform mixing of the nutrients, the stirrer speed was set at 150 rpm. 250 ml of the fermentation medium was used for experiments. Once the medium was transferred to the reagent bottle, it was autoclaved at 121 degrees Celsius for 20 minutes and cooled down. Nitrogen was flushed in the reagent bottles to ensure anaerobic environment in the reactor. Meghalophilus was inoculated in the sterile medium and the size of the inoculation was varied at 1 ml/L, 2 ml/L and 4 ml/L of the fermentation medium to study their effect on glycerol degradation. Studies carried out initially showed that crude glycerol volume of 15 ml/L is effective for hydrogen production. The bacteria also use visible light for fermentation. LED bulbs of 5W, 10W, and 15W were used to subject the photofermentation process to different light intensities. The gas produced during the photofermentation process was collected as a function of time to analyze the gas production rate and to determine the hydrogen yield. The residual glycerol in the fermented broth was determined by spectroscopic analysis. The gas collected was analyzed by gas chromatography with thermal conductivity detector. The flow process of the experimentation is shown in Figure 1. V. Results and discussion. The crude glycerol sample was analyzed by spectrophotometric method and the concentration of free glycerol in the sample was determined from the glycerol calibration chart. The glycerol content in the crude sample was found to be 0.58 mg/milliliter. 15 ml/L of crude glycerol was used as carbon substrate in the fermentation study. Therefore, the actual free glycerol available for bacterial fermentation was found to be 8.7 g/L. 1 effect of inoculum size. Inoculum size of meghalophilus was varied as 1 ml/L, 2 ml/L and 4 ml/L respectively. Hydrogen is produced as primary metabolite during the growth of the bacteria. Thus, monitoring the gas produced as a function of time, reflects the growth nature of the bacteria. The bacteria was found to have fast acclimatization to crude glycerol as the accumulation of gas could be noticed with respect to water displacement in the inverted cylinder with the inoculum size of 1 ml/L. The initial lag period was observed to be 3 days. It could be observed that as the inoculum size varied, the duration of lag phase in the growth cycle of the bacteria also varied. Increasing the inoculation volume helped to reduce the lag period. For inoculum size of 2 ml/L, the lag period was considerably reduced to 1 day and substantial increase was also observed in the volume of gas accumulated. The growth period was found to be longer than the lower inoculum size. For 4 ml/L of inoculation, the lag period was very negligible and the duration of gas generation was also observed to be shorter. The observations are shown in Figure 2. This showed that inoculum size has an impact on the bacterial growth and production of gas as its primary metabolite. With varying inoculum size, the substrate to inoculum size ratio was calculated and was obtained as 15, 7.5, and 3.75. It could be seen that the ratio decreased with increase in the inoculum size. The bacterial population in the fermentation medium was also larger as the inoculum size increased. Thus, the higher microbial population helped in faster bacterial activity that was conducive to reduce the lag period. This condition also resulted in rapid substrate consumption with the crude glycerol in the medium being limited to 15 ml/L. This could be related to the faster growth of the bacteria with 2 ml/L of inoculation. Thus, the rate of substrate depletion could have been higher with 4 ml/L of inoculation that led to shortened growth period and slowdown in the gas production. The substrate conversion was calculated with respect to the free and the residual glycerol in the fermentation medium. The amount of residual glycerol in the fermentation medium was determined to be 3.35 grams, 5.49 grams, and 5.81 grams and the substrate conversion was calculated to be 38.5% for 1 ml/L inoculum size, 63.10% for 2 ml/L of inoculum size and 66.78% for 4 ml/L of inoculum size. This showed that crude glycerol degradation was favored by higher inoculum size. The hydrogen content in gas samples from fermentation with 1 ml/L, 2 ml/L and 4 ml/L inoculum size were analyzed by GCTCD and was found that inoculum size of 2 ml/L showed highest hydrogen with 15%, V/V. Therefore, inoculation size of 2 ml/L was considered as effective for hydrogen production process. 2. Effect of light intensity. With inoculum size of 2 ml/L, found to facilitate good bacterial growth and hydrogen production, the photofermentation experiments were carried out at 3 different light intensities. LED bulbs of 5W, 10W, and 15W were used to supply light at varying intensities. The bacterium was found to be receptive of light source as it was found that increasing light energy helped to boost the bacterial growth. The growth period of the bacteria, for all the 3 light intensities, did not change from the growth period without light source. But a considerable difference was noticed in the volume of gas produced, with varying light intensity. When LED bulb of 10W was used, the volume of gas generated was found to be significantly higher than that generated at 5W. This was ascertained with the increase in the gas volume that was monitored over a period of time. As hydrogen is produced as a primary metabolite, the increase in gas volume shows that the bacteria uses light energy as additional energy source to break down the crude glycerol molecules in the medium. This in turn helps in better uptake of the nutrients, as a result of which, the volume of the gas produced also increased. In order to find the extent of gas production with increase in light intensity, 1,5W lead bulb used to supply the light energy for photofermentation. The volume of gas produced exhibited the same trend as that with the use of 10W lead source, with negligible increase in the gas volume, as shown in Figure 3. This implies that intensity of light with 1,5W lead source could have exposed the bacteria to excess energy which could have been detrimental, affecting the metabolic productivity of the bacteria. Thus, the volume of gas produced did not increase with increase in light energy. The volume of hydrogen collected with 5W, 10W, and 15W lead light sources were found to be 44.8 ml, 78.4 ml and 81.6 ml respectively. 3. Specific hydrogen production rate. The specific hydrogen production rate is defined as the volume of hydrogen gas generated with respect to the amount of substrate consumed per unit time of fermentation. The formula for specific hydrogen production rate is given in equation 1. The substrate conversion rate helps to analyze the rate of hydrogen gas generation and will be crucial data for designing a continuous fermenter. Specific hydrogen production rate. The volume of hydrogen produced with 1 ml/L, 2 ml/L and 4 ml/L inoculum size was found to be 26.4 ml, 73.6 ml, and 57.6 ml respectively. The specific hydrogen production rate was thus calculated to be 0.075 m3/kg.s, 0.139 m3/kg.s and 0.103 m3/kg.s. The highest specific hydrogen production rate was thus found to be for inoculum size of 2 ml/L. 6. Conclusion. The inoculum size of the bacteria and the light source of varying intensities showed that both of these can be crucial parameters that affect the substrate utilization rate and the hydrogen yield. It was found that increase in inoculum size led to better substrate utilization as the substrate conversion rate was found to increase with increase in inoculum size. The highest substrate conversion rate obtained with 4 ml/L of inoculum size with 66.78%. Even as the substrate conversion rate was higher for larger inoculum, it did not show similar trend for hydrogen yield. The yield of hydrogen was found to increase with increase in inoculum size from 1 ml/L to 2 ml/L while it decreased as the inoculum size was increased to 4 ml/L. This was due to the substrate depletion, caused with higher microbial population. Studies conducted with varying light intensity showed that at light energy supplied with 10W lead bulb, the bacteria were able to absorb light energy for effective production of hydrogen gas. The bacterial growth was observed to have slowed down with further increase in light energy, as excess energy led to bacterial growth inhibition caused by photo effect. This was correlated with respect to the gas volume produced, that did not show any significant variation compared to gas volume obtained with 10W light source. The parameters of inoculum size and the light intensity that could lead to effective crude glycerol degradation and yield hydrogen gas was determined to be 2 ml/L and 10W lead light source. Therefore, determination of photosynthetic active radiation is essential for effective utilization of light energy by photofermentative bacteria. 7. Acknowledgement. We thank the management of Dayananda Sagar College of Engineering and Ramaya Institute of Technology for their support and encouragement for the research work. We also thank Mr. Brijesh, young scientist, Biofuel Park, Gandhi Krishi Vinayana Kendra, UAS Bengaluru, and Dr. C.H. Sasi Kala, Professor and Head, Center for Environment, Institute of Science and Technology, Jawaharlal Nehru Technological University Hyderabad, for their help and guidance.